All right, so we are here to talk about photorespiration. And so we're going to have to do a little background first, and then I'll write out some things. All right, so let's just remind ourselves what Rubisco is and does. Rubisco is an enzyme. It stands for this right here, big name, that just means it adds a carbon dioxide to our UBP. So it is going to catalyze the reaction that joins these 5 carbons to these carbon dioxides to make the 6 carbon. All right, Rubisco is somewhat substrate specific, not exactly. And so what happens is Rubisco can bind CO2 to our UBP, but it can also accidentally sometimes grab an oxygen. So it's just kind of reaching out and grabbing, and whatever is closest is what it's going to grab. And so when our CO2 levels are high and our O2 levels are low, so there's lots of CO2s and very little O2s, when Rubisco reaches out and grabs, it's probably going to grab a CO2 because that's what there's more of. So just by nature of probability, it's probably going to grab a CO2. So it is going to take that 5 carbon, it's going to add the CO2, which is a 1 carbon, and that's going to make a 6 carbon that immediately breaks into 2 3 carbons. And that's what we want because they both go, they continue that process and end up down the road. So again, looking back at this, the 6 carbon breaks into 2 3 carbons, it gets all the energy. One of them leaves, yada, 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 and eventually this guy's going to make glucose. All right, so eventually we'll get to glucose. There's a bunch of other steps in there that I'm skipping, but just for simplicity's sake. So that's yay, Rubisco. That is what we want you to do. Good job, boy. Good job. All right, no Rubisco, when no Rubisco is being bad, is when our CO2 levels drop and our oxygen levels rise. Okay, this is going to happen when we close our stomata. All right, so why are we going to close our stomata? Well, we're going to close our stomata because it's hot. And when stomata are open, remember stomata are the little pores on the underside of the leaves, and their job is gas exchange. So their job is to bring in CO2 and for oxygen to leave, okay? But something else can happen too. When those stomata are open, water vapor can escape too, especially if it's really hot and really dry, so like in the summer or in the desert, okay? And so when they open their stomata, they can risk dehydrating if it's hot outside, so they might close their stomata to prevent dehydration. Well, when they close their stomata, they're still doing photosynthesis, so they're still producing oxygen in the light-dependent reactions. They're still using up CO2, right? They're putting CO2 into the light-independent reactions. So what you see happening when their stomata are closed, they're still producing oxygen, so those levels start to rise. They're still using up CO2, so those levels start to drop, and Rubisco just reaches out and grabs something, and it just happens to reach out and grab an oxygen, okay? And so it's going to join that oxygen to a 5 carbon. Well, oxygen is a no carbon, right? 0 carbon. And so when you add a 5 carbon to a 0 carbon, you still have a 5 carbon. That 5 carbon gets broken down into a 3 carbon. Yay, that's a smiley face. Sorry, I didn't do a good job. And sorry, that's my husband texting me. It gets broken into a 3 carbon and a 2 carbon, okay? This 2 carbon, not good, toxic, poison. If I could draw a skull and crossbones and you would know what they were, then I would do that. But if I drew them, you wouldn't know because I can't draw. But this is toxic, and so what the plant does is it has 2 options. It can either, wait for the bell, it can either keep this, which will poison it and kill it, so that's not really a good option, or it can send it to the mitochondria to be broken down in a kind of a abbreviated form of respiration, but that uses ATP, so it's going to waste some energy. So the plant can either die, or it can waste energy to break this down, which is not going to kill the plant, but it's going to be hard on the plant, right? We don't want to waste energy. That's not good for our bodies. And plants are the same way, but it's the better of the 2 options, okay? So this is called, this process of binding to oxygen, producing this 5 carbon that breaks into a 3 and a 2. The 2 carbon is toxic, gets broken down through an abbreviated form of respiration, is called let's put it up here, respiration. Photo because it has to do with photosynthesis, but respiration, whoops, I'm sorry, you can't see that. There you go. Respiration because it is involving the mitochondria and doing some parts of respiration to break down this 2-carbon molecule, okay, and burning up some ATP. All right, so I can define photorespiration, okay, so it is that process where rubisco binds oxygen to the RUBP, this 5-carbon, which was RUBP, creating this 2-carbon toxic molecule that has to be broken down. I can explain why it occurs. It occurs because rubisco is not perfect, and it does bind to both of these pretty much equally. So whichever one is in higher concentration is the one it's going to probably grab. So it just kind of reaches out and grabs. Now, if it reached out and grabbed a nitrogen, it's not going to do that. So it is somewhat substrate specific. It will really only bind these two. That I know of. So it's not going to just bind to any gas or any molecule, but unfortunately it can bind to oxygen just as much as CO2. So I can explain why it occurs, and that's just because those oxygen levels are higher than CO2, and because rubisco is not 100% substrate specific. I can explain why it is harmful to the plant because it takes this, or it produces this toxic 2-carbon molecule that will either kill the plant or to cause the plant to waste a little bit of energy to break it down. And I can explain why C3 plants are more likely to do photorespiration on hot, dry days. A C3 plant is a plant that does this normal reaction. And the reason it's called a C3 is because the first stable molecule that's produced after rubisco, so rubisco is kind of the starting point. After that, remember this one's super unstable, it's almost instantly broken, so it's not even around very long. The first stable molecule is a 3-carbon, so we call that a C3 plant. And they are more likely to do this because they don't have any defense against it. So when they get hot, they're going to close their stomata. When they close their stomata, their oxygen levels are going to rise and their CO2 levels are going to fall. And then rubisco is going to start doing the wrong job. So they don't really have a way to prevent it. And that'll make more sense if you understand C4 and CAM plants and how they do have a way to prevent it. But C3 plants do not. And so they're the ones that are more likely to do this photorespiration process on a hot, dry day because they're going to need to close their stomata to save water. Whereas C4 and CAM plants have other options, which we'll talk about later. So that is photorespiration in a nutshell. I hope that helps. If you have questions, let me know.